It is the top of the hour, so we'll get started. I'd like to welcome everyone to today's GlomCon seminar. I'm Dia Waguespack in Houston. Today our talk is Updates in Complement Biology and Health, Disease, and Therapeutics. Today we are very excited to have Dr. Alfred Kim.
All right, thank you so much. I'm going to go ahead and, yeah, perfect. Excellent. So I see some familiar faces, or at least names on the list. For those who don't know who of me, first of all, I appreciate the introduction, Dia. I've done a lot of work actually back in the past on podocytes of all things. I postdoced with Andre Shaw, but then once I actually started my own research group, I got very interested in complement, mostly through mentor of both Anuja Java's and mine, John Atkinson. And ever since then, I really got into trying to address some of the questions that I think one of the things in medicine we always, I'm surprised about is how many assumptions we use and they end up being right. But then when you actually start thinking about some of the things more research focused, a lot of these assumptions end up not playing out. And I think this is also true in complements. So while I'm going to be talking about a lot of aspects about complement from a principal point of view, of course, we're going to be talking about some aspects specific to glomerular diseases later on in the talk. So these are its disclosures. I don't think they're seeing me with this, but I just want to, for full transparency, I do a fair amount of consulting work with Exogen, which has a complement diagnostic product for lupus that's commercially available. And then I'm also chief medical officer of Kaifa, which is a kind of a newer biotech company in the complement diagnostic space. All right, so complement cascade, certainly one of the most ancient systems in the immune system. It's found in multicellular organisms as far back as the jellyfish, and it's also highly regulated. Most talks will include a figure such as this, showing layers upon layers of complement protein activation cascades. Of note, this is a review that Anuja led, discussing what we thought was the role of complement during COVID-19, early during the pandemic. But I get that when you look at figures like this, people don't really like complement that much, and I get that. I mean, when I was, again, doing my postdoc, the last thing I frankly wanted to think about was complement. Why? Because I just didn't understand it. I mean, this slide only covers actually about 60% of the entire cascade. So here are my goals today, and it's not to dive into every nook and cranny of complement. Again, this is going to be a more focused view about how the complement system can be perturbed by intrinsic and extrinsic factors to exaggerate complement function. And then that can then lead to a pathogenic phenotype associated with the disease. So we'll start off with a high-level overview, but this really lays the foundation for the discussion of several other diseases where complement inhibition has been successful, and how those data contribute to our understanding of how complement contributes to pathogenesis. Now, you'll see some diseases on this list that nephrologists and rheumatologists do not treat, but I'm offering these examples in order to introduce to you the kind of the conceptual proof of principles that apply to complement-mediated diseases and that can be treated by both nephrologists and rheumatologists. There are certain diseases here I'm not talking about, C3G, I won't be talking about explicitly, but I will be talking about iptacopan. So it's ironic that obviously Novartis is sponsoring this. And the part of the reason why I can't cover every disease is that virtually every pathway has some experimental, i.e. animal model data or biomarker data in humans of virtually every disease. And I'll talk about this in a couple slides. So again, I'm not going to be talking about every single complement, the role of complement in a wide variety of renal conditions, but I will bring up IgAN because this is a very confusing condition to me, and I'm not sure the explanation in the textbooks or in reviews about what complement is doing there is exactly right. Also, at the end, I'll be taking a little bit of a deeper dive into some more speculative mechanisms that we think is going on based on our own unpublished data as applies to lupus and probably has implications of the ANCA-associated vasculitis and possibly IgAN. So again, this is where the discussion will move into a space where clinical actionability may not be immediately clear, but I want that discussion just to serve as a sneak peek into the concepts of complement biology in our diseases that may evolve in the future.